Hello, everyone. So first of all, I would like to thank the organizers for inviting me to this great event and to this, learn more about this great initiative. And I'd like to thank Tomas for excellent in-depth introduction to X-ray CT scanning, which makes my task very easy because what I'm going to do, I'm going to give you just some hopefully inspirational examples of what can be done if you have CT data and soil data. And my examples will be much more limited than what the potential and breadth of data that MONet project will offer. So you have much more capabilities than what I will be talking about. I'm a big fan of X-ray CT, and the reason why is because it offers a great opportunity of studying soil as a holistic functioning body. And analogy that I like to think of when I'm thinking about soils and CT is analogy with a human body. Just like our bodies consist of tissues and vessels through which all the necessary flows necessary for our functioning taking place. Just the same in soil, it's a combination of solids and voids, soil pores, are in essence the vessels through which all the fluxes take place and where the soil biota and microorganisms live. One opportunity that X-ray CT offers is to study that system of vessels in its intact shape. So if we look at something as in a holistic way, like a soil, and what we typically do, what soil scientists typically do, we take soil sample, we sieve it, we grind it, we do some measurements. Well, it's just like, okay, is that the one thing? It's in essence just like demolishing a building and then trying to infer how that building functioned based on that pile of stones and debris that came out from it. And until relatively recently, working with those seed soils was the only option that soil scientists had. But fortunately, with development of X-ray CT and big progress in it, it became possible for us to look at soil structure and soil pores in their intact shape, even though we're looking at fairly small, intact soil samples, still they are intact. So first applications of X-ray CT in soil sciences, people were using medical scanners, and there are still some soils groups that use those with doing quite successful work. But as Tomas was saying, a lot more can be done with synchrotron facilities, where you get nice monochromatic beam that allows you to get images of very high quality with very high resolution. And a lot can be done now with benchtop scanners like the one that you have here. That's the one we have access to at Michigan State University. And Michelle is a great colleague who was most instrumental to collecting the data that I will, the scanning results that I will be showing. And again, it's I think it's so wonderful that MONet is considering and using this scanning and looking at pore structure in intact shape because I think this is ultimately the right way to proceed. So as I said, I will be talking about some examples of using X-ray CT scanning for soil pore characterization. And what I have here is the list of the characteristics of the soil pore system that commonly seen in X-ray CT studies, what people commonly work with and use, and well, that's also happened the ones that we use in our lab. Plus, of course, besides all those pore characteristics, they're particulate organic matter and plant residues. You've heard a couple of times today about this serious set of steps that need to be taken place before one has the actual images, or not images, but actual data from the X-ray CT scanning. So after you scan, you have to work with the image to make it nice, reduce noise, filter, whatever you have. You need to segment it. You need to identify pores. If you are interested in pieces of organic matter or residues, you need to identify and segment those. After that, you need to work with tools to generate pore size distribution, so to characterize those pieces of organic matter. One thing I want to bring to your attention is that it's one of the limitations, or at least what we encountered as a limitation in working with X-ray CT, is that each of these steps, even though for every image analysis process for every soil sample, these would be the steps. Yeah, these are the steps. That's set. But each step involves a very long list of settings, parameters, and it might be they might not be the same, or I should say they will not be the same for every scanning setting. They might not be the same for every soil. They might be different for soils of different textures, soils of different mineralogies, and even for soils that same texture, same mineralogy, but with different vegetation or management history. So identifying, finding the proper settings for cleaning, for filtering, for segmentations, that's always been a struggle for us to making the perfect decisions. That's why, again, I think it's you're so lucky to have Tomas and his team to do this very important and very sometimes very hard work for you. And again, it is important because what you get as far as the data very heavily depends on the decisions that are made during filtering and segmentation stages. Okay, so let me just go through some of these one by one. So image-based porosity, that's just what the volume of the pores that we can see on the image, and of course that's a function of the resolution. And then the distribution of the pores that we see by size of the pores that we see at that resolution. I'm not going to talk much about the image-based porosity, and that's just because in experience of our lab, we rarely find it sensitive enough or interesting enough to do anything with. So it's probably soil specific and test specific, but for us, it never really was a very useful parameter. So I'll talk about pore size distributions. And as an example, excuse me, I'm going to tell you a little bit about the work that PhD student in our lab, Jinho Lee, did as part of his dissertation. So he worked with soils collected from 6 experimental sites going through all the way north to south in Michigan and Wisconsin. And at every site, there is an experiment there with replicated systems for restored prairie and monoculture switchgrass. So he, at those replicated plots, he collected intact soil cores, 5 centimeter in diameter, subjected them to scanning. And here are just some of those image processing and scanning elements, some of what you've heard from Tomas about. So the scanning resolution was 18 microns. Jinho wasn't happy with just using a single thresholding method for solid segmentation, so he used 8 and then averaged what he got, and that's how he got his pore size distribution. So what you see here is his overall pore size distribution data, the averages for his multiple sites and switchgrass and prairie vegetation. On the x-axis are pore diameters, on the y-axis are the volumes of the pores of these different sizes. So that would be very typical of how one would present the pore size distribution results from a CT scanning. One thing, as far as sort of like what we learned or what something to keep in mind is another complication of that, I should say, well, something one has to keep in mind when working with CT data and pores or pieces of organic or whatever you want to identify on the image is that even though your scanning resolution, like in our case, is 18 microns, what we can realistically identify and work with, those objects that we can realistically identify, they are at least twice that resolution, or better, they are 4 or 5 times bigger than the resolution. So why I'm saying this when showing you this graph is because you're probably looking at the sour volume of pores of the smallest size. This is our resolution, and you might think, well, that's a very strange looking pore size distributions that these people got. How come they have so few of these small pores? This is an artifact because our resolution is 18, so we hardly see any real pores at that particular resolution. The next one, 50, it's better, but it's probably also underestimated. It's that 100 is, yeah, definitely we are certain about that. Excuse me. So what Jinho did in this study, he specifically focused on the pores of this size range, 50 to 150 micron in diameter. And the reason for that is our past work and a lot of literature demonstrated that pores of this size range very often they are associated with the most of the soil respiration, the strongest decomposition of the plant residues, the highest microbial activity. So he specifically focused on that particular range. So also want to bring to your attention these sample images on the bottom. What you see there, the all visible pores, and in light brown, those medium-sized pores, those 50 to 150. And on the left, in each pair, the left one is from his coarse textured or fine textured site. And on the right are the ones from his coarse textured site. And what you see is a big difference due to texture, because yes, texture is the most important factor that influences the volume of these pores in the soil. But besides texture, the vegetation history, prairie versus monoculture, also had a minor but consistent difference. So what you see here on the x-axis are the individual sites arranged in this from finest silt loam to sand on the right. And on the y-axis are the volumes of pores in the size range. Blue is prairie, orange is switchgrass. And you can see that consistently we have this great volume of these pores being developed after those 8 to 10 years during which the experiment with prairie versus switchgrass vegetation was in place. So I didn't tell you, I should have told you from the beginning that the goal of Kravchenko's study was to see what role soil texture and soil structure, that is pores, what role they play in promoting soil carbon gains. So with that goal in mind, he combined his findings in terms of these mid-sized pores with his findings on the differences in soil organic carbon between these two systems. And the key thing that you might notice here is just the same as with those pores in every site. The blue bar, the prairie, is higher than the switchgrass. So prairie was more effective in increasing soil carbon, resulted in higher carbon in all these very different soils than the switchgrass. And the two were closely related. Here, the more medium-sized pores were generated with prairie vegetation as compared to what was generated with switchgrass vegetation. The more where the bigger was the difference between the two systems in terms of soil carbon. So that allowed Kravchenko to then proceed with testing a number of interesting, quite complex hypotheses that he built as far as how texture drives pores, drives labile carbon and microbial activities, how all this contributes to soil organic carbon. So yeah, that I'm not going to get a lot into that because I could talk for a couple of hours just about that. Okay, so another example, and this time this would be this other type of characteristics that you can get from pore size distribution, from pore data from the image. And this is at the moment my big favorite. So that is the distance from pores to solids. So you can think about it as a measure of the spatial variability, spatial distribution of pores within the soil matrix. If the pores are like everywhere uniformly distributed, you would expect that the distance is small from any point in space in the soil matrix to the pore. If you have one big pore and nothing else for, in your entire pore, then you expect that that distance would be quite high. What I have here is 5 recent studies where different groups, including ours, used this particular criterion to predict nitrous oxide emissions and denitrification. So yeah, I'm very fascinated by all of them very much. Just as an illustration, so this set of images is from this paper by Roche from Stefan Schlutter's group in Germany. So on the left, you see the CT image of the soil that they worked with. So gray is soil, black and grays, light grays are water and pores. In the middle is the same image where they segmented the solids, which is still gray here, and the pores. And what they did further, they classified the pores depending on whether or not they were connected to the atmosphere. The red ones are the ones connected to the atmosphere. The pink ones are not connected to the atmosphere. And the blues are the pores filled with water. So then the leftist, well, the rightist for you image, the multiple colors represent the distances to those pores connected to atmosphere, or what you could use as a measure of how anaerobic each particular area of that soil pore was, which these researchers then found was a very nice predictor of how much nitrous oxide was emitted and what the denitrification was. Another very brief example from our lab. Again, it's the same experiment as the one I told you about that Kravchenko was on. It's restored prairie versus monoculture switchgrass. And these two colored image examples represent the pores. So the black areas with those yellow outlines, these are the pores visible at our image resolution. And well, with resolution of 18 and pores that we see basically some 36 micron diameter, these we can realistically say, yeah, these are would be typically air-filled pores. And the colors, they represent how far, they represent those distances. How far is the soil matrix from those pores? The blues, it means that soil matrix is close to the pores. Oranges and whites are the soil matrix that is far. And what you see here is what we just typically seen there. That in switchgrass, the distances to those pores, to the pores, were bigger than in prairie. And as I told you earlier, we consistently see in these systems that soil carbon is higher in prairie than in switchgrass. So how these two might be related, those distances to pores versus soil carbon, well, if you think about pores of this size range, they are the ones through which carbon comes in. This is where the fine roots are, root hairs are. This is where the fluxes of soil solution with dissolved organic matter, where it fast comes through. So they are the sources of carbon, and they are also the location where microbes tend to live. So what we think is happening is something like this, that if, let me start from the one on your right, if those pores, as they are in prairie, nicely spread through that entire soil matrix, and that carbon comes in and microprocess it, and those processed carbon diffuses from the pores into surrounding soil matrix, it just gets everywhere. But if the pores are very far from each other, then what we think is happening is that there is this substantial body of soil matrix that could protect that carbon, but because it's just so far from any pores, it doesn't get there. So again, just a short example. Another, let me just very briefly, think I'm running short on my time. So another example is what could be gained from that information is the shape of pores. And specifically what you can get from pore shape is you can infer their origin. So biopores, pores of biological origin, something made by roots or by earthworms or by both, is what you would expect to be of nice cylindrical shape. Cracks of non-biological origin is what you would expect to have all kinds of shape. So Mike Lucas is the person who pioneered, explorations of these biopores, identification of them, and specifically in the work that he did when he visited our lab for a year as a postdoc. He explored how different cover crops impact development and reformation of these biopores. So he worked with these 5 different cover crops that we had in these small demonstration cover crop demonstration plots. We had soil cores taken same size as before, all the same settings, and we had scanned them. And then what Mike did, next season, he put them back in the ground. Well, he wrapped them in a mesh that would be penetrable to the roots. So he buried them under the ground in the plots of the same cover crops from where they came. So as a result, he had tons of variety of image analysis done there. But what, again, something that I haven't mentioned before is what he could do. He could look at the biopores and their size distribution, but he also could look at what happens with those biopores. And this figure from his paper described that. So the top bars in multicolors are the biopores that were generated after that season on the ground, in the ground. The bottom gray ones are the biopores that disappeared. So again, I'm running out of time. So just one point I want to mention relevant to using the images and the information that you can get from them. So, and this, for this one, so I brought to your attention, the dangers and inconsistencies that we might get on a very fine size of the pores that we work with. But here I want to bring to you the other side. So you might be wondering about this very right part, that is the biggest biopores, the ones that are 2 millimeters in diameter or greater. And you can see that we don't have any statistically significant differences anywhere there. And you might be wondering how could that be, because they are the ones that probably are most heavily used and probably should be, moved around and changed most intensely. And I agree with you. So the reason why we're not seeing that is because of our sample size. So in our 5 centimeter pores, we just don't have enough representative large biopores. And so we have 1 or 2 here and there, and they're just not, we're just not able to accurately and certainly estimate and quantify them. So, then he went into this crazy thing, trying to explore what could be the causes, who is destroying those biopores. And, he looked at the images and identified those causes as either earthworm casts or excrements of some insects. Don't ask me how he did that. I don't know. But then, that was a lot of exciting discussions we had about that. So my last example is, again, Mike's work. And in this study, it's particulate organic matter and pores that he combined to predict field emissions of nitrous oxide. So he worked with this replicated site with specifically with these three corn, sorghum, and switchgrass systems. And during the season, he had the gas chambers installed there, and multiple times during the season, he was doing greenhouse gas measurements. And then at the end of the season, he took those same old intact soil cores from the locations of the chambers and scanned them as usual. So what seemed to be the best predictor out of a large number of pore characteristics and variety of characteristics that he got from those images, what was plus, yeah, definitely, chemical characteristics and water content and soil nitrate. So what worked the best was a combination of particulate organic matter information from the images with a characteristic of where those pieces of particular organic matter were in the soil matrix. That is combination of that home images with pore images. So the poem that was located far, that was basically according to our distance to pores characterization, the poem that was located in anaerobic, relatively anaerobic condition. So that those fragments that were again in anaerobic conditions, those fragments were the best predictors we got as far as how much nitrous oxide and denitrogen came out during the season from the field and what was the fraction of bacterial denitrification there. And if you think about those fragments of foam in those relatively anaerobic conditions as hotspots of nitrous oxide production, then it makes perfect sense. And with CT scanning, one can actually quantify and understand that. Okay. Let me just skip this. Couple of things. So those soil pores and organic residues, that's what we in our lab work primarily with. As was discussed, roots can be very nicely visualized, and there are a number of research groups across the world who kind of made it their business to do it very effectively. You can see water, even though that is a tricky business. You can, I don't think you really can see, or nobody was very successful in seeing soil organisms in the soil, but outcomes of activities for some of them, like earthworm burial systems and what happens with them. People have done very interesting and exciting work with that. So I just wanted to make sure I mentioned those examples. Okay, and so that brings me to my just a summary that soil is so functioning means looking at the soil as one whole living ecosystem, and we can only appreciate that functioning if we keep it as close intact as possible. And it's great now that we have X-ray CT that enables us to do that. So just acknowledge my research team and funding sources, and I don't know if I have time for questions. So what's the plan?
That was a great presentation, putting everything in perspective. Kamas had posted a question in the chat. He asked whether there was any staining used to light up the POMs for the CT.
No, in these studies, no, we haven't used any staining. We just try to work with raw images. Yeah, but yeah, we dabbed a little bit with iodine and osmium staining, and it for particular organic matter, it can work very nicely and make one's life easier. One reason why we haven't done a lot of that is because very often what we do with our samples, we subject them to all kinds of incubations and subsequent experiments that involve microbial activity. And if you add osmium, it just kills, so iodine, they just kill everyone. So those, I would see them as sort of the very last step of whatever experiments you might want to be doing.
Thank you. We will take a quick break, and for everybody that is here in person, please chat with Sasha and ask her questions about this break.